In 1935, a marine biologist named James McMichael was walking along a beach in Queensland, Australia. He found a beautiful cone-shaped shell lying on the sand near the water. What he had found was a Conus geographus, the geography cone snail. It is one of the most visually distinctive and beautiful shells found in any ocean. He picked it up the way any naturalist would pick up a remarkable specimen. The snail was still fully alive and active inside that shell. It stung him through his palm with a harpoon he never felt entering his skin. Within 5 hours of that single contact, James McMichael was dead. He was a professional marine biologist who spent his career studying mollusks specifically, and he died from picking up a shell he found beautiful on a beach. Today we are talking about cone snails, specifically the venomous cone snails of the family Conidae, a group of animals that look like collectible decorative objects and function as some of the most sophisticated chemical weapon systems in the natural world, and whose venom contains compounds that are currently being developed as pain medications, in a 1,000 times more potent than morphine without causing addiction. Let's start with what a cone snail actually is, before we get to what it does. Cone snails are marine gastropods, snails that live in the ocean. There are approximately 700 to 900 known species of cone snail distributed across tropical and subtropical oceans worldwide. The majority live in the Indo-Pacific region, the waters around Australia, Indonesia, the Philippines, and nearby areas. They range in size from under a centimeter to over 20 centimeters in shell length. They live on coral reefs, sandy bottoms, and in shallow coastal waters. Most species are active primarily at night and spend daylight hours buried in sand or hiding under coral. During the day, they look like nothing more than an attractively patterned shell lying on the seafloor. And this is the first and most important thing to understand about the danger they represent. The danger a cone snail represents is not visible from the outside of the shell. A venomous cone snail looks identical to a harmless, collectible shell. The same shape, the same patterning, the same visual character that has made cone shells prized by collectors for centuries. European shell collectors in the 18th and 19th centuries paid enormous sums for rare cone shell specimens. At one point, a particularly rare Conus gloriamaris, the glory of the sea cone, sold at auction for more money than a painting by Vermeer that was sold at the same auction in the same year. The shells were that beautiful and that coveted by collectors. And a significant portion of those collectors had no idea they were handling animals that could kill them in under an hour if handled incorrectly. Now, let's talk about the weapon, because the cone snail's delivery mechanism is unlike anything else in the animal kingdom. Most venomous animals deliver venom through a fixed bite or sting. A snake strikes forward with its head. A bee drives a stinger in a single direction. A spider bites from the front. The cone snail has a radula, a modified tooth structure connected to a venom gland via a highly extensible and directionally flexible muscular tube called a proboscis. This proboscis can extend in any direction from the snail's body. Forward, backward, sideways. The cone snail can sting in any direction, including directly backward toward a hand holding it. The proboscis moves fast enough that the initial penetration is often not felt at all. The harpoon-like radula tooth penetrates skin, injects venom, and withdraws in a single rapid motion. Multiple accounts from people who survived cone snail stings describe feeling nothing initially, and only realizing they had been stung when they noticed a small puncture mark or felt the first symptoms beginning. The venom itself enters the bloodstream almost immediately after injection, and the first symptoms can appear within minutes of the sting, depending on the species and the dose received. Now, let's talk about the venom itself, because this is where the cone snail becomes genuinely extraordinary as a subject. Conotoxins, the venom compounds produced by cone snails, are not a single substance. They are a complex cocktail of hundreds of different small proteins called peptides. Each cone snail species produces a different specific combination of conotoxins. A single cone snail venom can contain between 50 and 200 distinct peptide compounds simultaneously. and researchers studying cone snail venoms have identified over 100,000 different conotoxin compounds across the approximately 700 to 900 currently described cone snail species, making cone snail venom one of the richest single sources of pharmacologically active compounds on Earth. These peptides work by targeting specific ion channels and receptors in the nervous system, each one with extraordinary biochemical precision and receptor selectivity. Each conotoxin peptide targets a very specific type of receptor or channel, blocking or modifying its function in ways that interfere with normal nerve signaling. The collective effect of hundreds of these peptides acting simultaneously on multiple nervous system targets produces the rapid paralysis and neurological shutdown that makes cone snail venom so lethal. The primary prey that fish-hunting cone snails target is exactly that, a fish. Fish are fast, mobile animals that would easily escape from a snail that had to physically catch them. The cone snail solves this problem by shooting its harpoon into a nearby fish from a distance and delivering a venom that immobilizes the fish almost instantly before it can swim away. The speed of action required to catch fish prey has driven the evolution of conotoxins toward extreme potency and extremely rapid onset compared to venoms evolved for defense rather than prey capture. This is why cone snail venom acts so quickly compared to most other animal venoms. It evolved specifically to work fast enough to stop a fish from escaping, and when it enters a human nervous system, it works with the same speed against a system it was not designed for. Conus Geographus, the geography cone that killed the marine biologist in 1935, is the most dangerous species. It is a fish hunter, and its venom is calibrated to stop fish movement within seconds. In documented human cases, the venom from a geography cone can cause paralysis of the extremities within minutes, followed by respiratory failure, vision impairment, and cardiovascular effects. Death can occur in as little as 30 minutes to a few hours without immediate medical intervention, and there is no antivenom available for any cone snail species. This means that for the most dangerous species, there is no specific medical treatment. Patients who survive serious cone snail stings survive through supportive care, artificial ventilation to maintain breathing while the venom effects pass, and monitoring to manage cardiovascular and neurological complications as they develop. The survival rate from Conus Geographus stings without medical intervention is estimated at around 70% fatality, making it one of the most lethal animal venoms on Earth measured by untreated outcome. And this animal lives in shallow reef waters around popular tourist beaches. All of this in shallow reef waters in one of the most visited ocean regions on Earth, and is routinely picked up by tourists who mistake it for an empty decorative shell. Now let's talk about the pharmacological side of the cone snail story specifically, because the exact properties that make conotoxins so dangerous to humans also make them extraordinarily interesting to medical researchers. The extreme selectivity of individual conotoxin peptides is the key property. A typical pharmaceutical drug acts on a broad class of receptor or target and produces therapeutic effects alongside a range of side effects caused by that broad action. A conotoxin peptide evolved to block one specific subtype of one specific ion channel, with almost no action on any other receptor in the nervous system. This specificity is something pharmaceutical chemists work extremely hard to achieve in synthetic drugs, and almost never achieve to the degree that evolution has achieved it in these small peptides. Researchers began systematically studying conotoxins for medical applications in the 1970s and 1980s, and the first conotoxin-derived drug to reach clinical use was ziconotide, approved by the United States Food and Drug Administration in 2004. Ziconotide is derived from a conotoxin found in Conus magus, the magician cone snail. It works by blocking a specific calcium channel in the spinal cord called N-type voltage-gated calcium channel. This channel is specifically involved in transmitting pain signals from peripheral nerves to the brain. Blocking it reduces pain signal transmission without affecting other nervous system functions. In clinical trials, ziconotide demonstrated efficacy for severe chronic pain, particularly in patients where opioid medications had failed to provide adequate relief. The drug is estimated to be approximately 1,000 times more potent than morphine by weight. And critically, it does not act on the opioid receptors that generate tolerance, addiction, and withdrawal. Patients who take ziconotide do not develop the opioid tolerance cycle that makes long-term morphine use progressively less effective over time, and they do not experience the withdrawal syndrome that makes opioid discontinuation so difficult. Ziconotide is currently used for patients with severe intractable pain who have not responded to other treatments. It requires intrathecal delivery, injection directly into the spinal fluid, because it cannot cross the blood-brain barrier when taken orally. This delivery requirement limits its current use to specialist pain clinic settings, and researchers are actively working on delivery mechanisms that would make it more broadly accessible. But as a proof of concept, it established that cone snail compounds can produce clinically useful drug effects in humans, and opened a significant research program aimed at finding additional therapeutically useful conotoxins. Researchers studying conotoxins have now identified compounds with potential applications, not just in pain management, but in cardiovascular disease treatment, neurological conditions including multiple sclerosis and epilepsy, and certain aspects of cancer pain and progression management. The breadth of potential applications reflects the breadth of nervous system targets that conotoxins have evolved to modulate, because the nervous system of a fish is mechanistically similar to the nervous system of a human being. The tools evolution developed to stop a fish can be adapted to manage human neurological conditions, with sufficient understanding of which specific peptide targets which specific receptor. Now, let's talk about how cone snails actually hunt prey in the wild, because the hunting behavior is as unusual as the weapon they use to complete it. There are 3 broad hunting strategies across the cone snail genus. Fish hunters like Conus, Geographus, and Conus striatus extend their proboscis toward a resting fish, deliver a rapid strike before the fish reacts, and immobilize it with fast-acting venom. The fish is then engulfed whole by the snail's expandable mouth. The engulfing process takes time, and the cone snail's expandable mouth can stretch to accommodate fish, substantially larger than the snail's shell opening would suggest possible. Some fish hunter species use a different and even more unusual approach. They produce venom containing insulin-like compounds and release these into the surrounding water. The compounds cause hypoglycemia, dangerously low blood sugar, in nearby fish. Fish experiencing hypoglycemia become sluggish and disoriented before the snail has even struck, pre-stunning prey with a chemical released into the water before the harpoon is deployed. The second group are worm hunters, cone snails that specialize in hunting polychaete worms. These species are generally less venomous than fish hunters because worms are slower prey that do not require instant immobilization to catch successfully. The venom of worm-hunting cones is still pharmacologically complex and interesting to researchers, but the clinical danger to humans is significantly lower than fish-hunting species. The third group are mollusk hunters, cone snails that eat other snails and mollusks, including in some cases other cone snails of different species. These too carry venom, but their strike mechanics are typically oriented downward into prey, rather than extensible in any direction the way fish-hunting species have evolved. The directional flexibility of the proboscis appears to be most extreme in fish-hunting species, where the ability to reach prey that approached from any angle was under strong selection pressure. This has direct implications for human safety, because fish-hunting species are the most dangerous to handle, and the most likely to sting in the backward direction toward a hand holding the shell. Now, let's talk about how researchers actually study cone snail venom in practical terms. Because collecting and analyzing these compounds is genuinely difficult work, cone snails must be collected alive and kept under controlled conditions for venom extraction. This involves handling animals that can sting through collection gloves of inadequate thickness, and through shell openings that researchers sometimes do not correctly anticipate. Multiple researchers studying cone snail venom have been stung during specimen collection. None of the documented researcher stings from fish hunting species resulted in fatality, but this reflects careful, immediate medical management rather than the inherent safety of the exposure. The venom extraction itself requires specialized techniques to collect the intact peptide mixture, without degrading the compounds that make the venom pharmacologically active. Once extracted, individual peptide components must be separated, and each one characterized individually. With over 100,000 known conotoxin compounds across all species, and more being identified as new species are described, or existing species are studied more thoroughly, the catalog of known conotoxins represents an enormous research opportunity. The vast majority of identified conotoxins have not yet been fully characterized pharmacologically, meaning we do not know what specific target each one acts on, or what medical application it might have if that target turns out to be therapeutically relevant. Researchers describe the total conotoxin library across all species as an essentially unmapped resource with enormous potential clinical value. Each new species characterized and each new peptide isolated represents a potential drug candidate that required hundreds of millions of years of evolutionary refinement before any pharmaceutical chemist anywhere in the world began working on them. The scale of the opportunity is directly connected to the scale of the difficulty. Extracting and characterizing peptides from living animals that can sting researchers, that can kill in under an hour if the wrong species is handled incorrectly, while working through an identified library of over 100,000 distinct compounds. With the current research capacity and funding levels available to this field. Now, let's talk about the ecological role cone snails actually play in reef ecosystems. Because beyond the venom and the pharmacology, they are functional components of the systems they inhabit. Cone snails as predators help regulate populations of the fish species and worm species they target on coral reefs. Fish hunting cone snails selectively remove fish individuals that come close to the substrate, or rest near coral surfaces in ways that place them in range of a cone snail strike. This selective predation may have subtle effects on reef fish population structure and behavior over time. Researchers studying coral reef ecosystems have found cone snail populations to function as reliable biological indicators of overall reef ecosystem health, because cone snails require specific prey populations and specific substrate conditions to thrive. A reef with declining cone snail populations often shows other signs of ecosystem stress, particularly in the fish and invertebrate populations that make up their prey base. The shells of dead cone snails also serve as housing for hermit crabs and other reef animals that depend on suitable shell structures for protection while they grow. The same beautiful shells that collectors prize and that make living specimens look harmless become critical infrastructure for other species after the cone snail itself has died. Now, let me return to the specific place where this story began, to that specific Queensland beach in 1935, and to James McMichael standing on it, to a professional scientist who knew what he was looking at and picked it up anyway, because the shell was beautiful and the animal inside it was invisible. This exact sequence of events still happens today in those same coastal waters. Emergency room records from hospitals near reef areas across the Indo-Pacific contain cases every year of tourists and snorkelers presenting with cone snail stings, from animals they picked up from the seafloor or from the beach. In many of these cases, the person did not know the animal was dangerous. And in a smaller number of cases, they knew in general terms, but did not connect the knowledge to the specific shell in their hand. The knowledge that cone snails are dangerous is not widely distributed among beach-going populations. In the reef regions where cone snails are commonly found on the seafloor, the distinctive conical shape that makes them recognizable to shell collectors is not a recognized danger signal for most people who encounter them. A tourist snorkeling on a reef in Indonesia or the Philippines, bending down to pick up an unusual shell from the sandy bottom near coral, is not typically running through a mental checklist of venomous invertebrates before making that decision. They see something visually striking and they reach down and pick it up. which is the same decision James McMichael made in Queensland in 1935, and which continues to be made regularly in the same waters by people with no way to know better. Write in the comments if you have ever picked up a shell without checking whether it was alive, because the honest answer for most people who spend time near tropical reefs is very probably yes. And the difference between a harmless experience and a medical emergency in that moment is which specific shell among many was the one you happened to choose. There is a specific historical incident involving cone snails that most people have never heard of, and it illustrates the danger more vividly than any abstract description of venom potency can. In 1989, a 44-year-old man in Hawaii picked up a textile cone, Conus textile, from a shallow reef. The textile cone is one of the more visually striking species with an intricate brown and white pattern. He placed the shell, with the snail still alive inside, into his shirt pocket. The cone snail stung him through the fabric of the pocket while he was walking back toward the beach. He felt only a slight tingling sensation at the contact site initially. By the time he reached his hotel room, he was experiencing numbness in his hands and legs. He was transported to a hospital, but despite aggressive supportive care, he died within hours. The textile cone is not the most dangerous species. That distinction belongs to the geography cone. But it is dangerous enough to be fatal, and its shell is beautiful enough to be routinely collected by beachgoers. The sequence of events in this case, pick up, carry, and pocket, get stung, die, represents the specific scenario that researchers and emergency physicians in reef-adjacent areas consider the primary public health concern. Not deliberate handling, but incidental contact with a shell that was assumed to be empty or harmless. The shell he had placed in his pocket on the way back from the reef was not empty. It had been a living, venomous animal from the moment he found it on the reef floor. It was exactly what it appeared to be, a living animal with a weapon it would use if threatened. And being placed in a shirt pocket is a significant threat from the cone snail's perspective. Now let's address something that connects the cone snail's venom to a much broader story in medicine. The development of ziconotide was the beginning of conotoxin-based medicine, not the end of it. Since ziconotide's approval in 2004, researchers have identified dozens of additional conotoxin compounds, with potential therapeutic applications across multiple disease areas. One area that has attracted significant research attention is the use of conotoxins in treating certain types of epilepsy. Some conotoxins target sodium channels in neurons in ways that could reduce the abnormal electrical activity that generates the abnormal electrical activity responsible for seizure events. Current anti-epileptic drugs act broadly on neuronal activity and cause significant side effects, including cognitive impairment, sedation, and in some cases mood disruption. A conotoxin that targets specifically the sodium channel subtypes involved in seizure generation, without affecting others, could theoretically provide seizure control with substantially fewer side effects. This is still primarily laboratory and animal model research, but the specificity of the compounds makes them genuinely promising candidates for this application. Another active research area involves the use of conotoxins in treating nicotine addiction. Certain conotoxins block nicotinic acetylcholine receptors, the same class of receptor that nicotine acts on. This creates the theoretical possibility of blocking nicotine's rewarding effects in a way that could support smoking cessation without requiring the patient to fight pure willpower against a fully intact and operational neurological reward signal firing normally. Conotoxin compounds in this category are in various stages of preclinical and early clinical research. None have reached the same stage of development as ziconotide as of the most recent published literature, but the scientific rationale is established and the animal study results are generally supportive of further investigation. There is also work being done on conotoxins as potential treatments for cardiovascular conditions. Certain peptide compounds from cone snails act on potassium channels involved in cardiac rhythm regulation. Irregular heartbeat, arrhythmia, involves abnormal cardiac electrical activity, and the same channel-specific selectivity that makes conotoxins interesting for pain and neurological applications, makes them potentially useful for restoring cardiac rhythm without broadly disrupting heart muscle function. Cardiovascular applications remain further from clinical use than pain management applications, but they represent part of the broad landscape of what researchers believe the conotoxin library may eventually offer. The scope of this research program is directly connected to how many different venom compounds exist. Across the full range of 700 to 900 cone snail species producing distinct compounds, a 700 to 900 species library, each producing 50 to 200 distinct peptide compounds, represents an enormous number of potentially unique molecular tools. Most pharmaceutical drugs currently in use were derived from or inspired by natural compounds, aspirin from willow bark, antibiotics from molds and soil bacteria, antihypertensives from snake venom. The history of pharmacology is largely the history of identifying what evolution already figured out, and finding ways to apply those evolved solutions to specific human medical problems. Conotoxins represent one of the largest untapped libraries of evolutionarily optimized molecular tools, still waiting to be fully characterized and assessed for clinical relevance. This is one of the more striking arguments for biodiversity conservation in purely practical human terms. The cone snails that live in the reef ecosystems currently under threat from coral bleaching and ocean acidification carry within them pharmacological tools that no human chemist could have designed from scratch. Losing a species before its venom compounds have been characterized represents the permanent loss of molecular tools that evolution took hundreds of millions of years to develop. A potential drug that might have addressed a specific type of pain or neurological condition, for which no currently approved drug provides adequate relief for many patients. Gone permanently before any researcher ever isolated or characterized it. Now, let's talk about the taxonomy of danger within the cone snail genus. Because the public health risk is not uniformly distributed across all 700 to 900 species, roughly 30 to 40 species are considered potentially dangerous to humans, with perhaps 15 to 20 representing a serious risk of fatality if a significant sting is delivered without medical intervention. The remainder of the genus, the majority of species, are primarily worm hunters, whose venom is pharmacologically interesting, but not calibrated to produce rapid vertebrate paralysis. The most dangerous species are predominantly found in the genus Conus proper. And within that genus, the fish-hunting species represent the highest risk category. Conus geographus is considered the most dangerous with an estimated pre-treatment fatality rate around 70%. Conus textile, the species involved in the Hawaiian fatality described earlier, is also highly dangerous. Conus tulipa and Conus striatus round out the group most frequently associated with serious human envenomation. All of these most dangerous species are among the most visually striking in the genus. All of them are found in shallow reef areas accessible to snorkelers and waders, and all of them look similar enough to genuinely harmless cone species. That visual identification without expertise is not a reliable safety strategy. Researchers and reef safety organizations consistently give the same advice. Do not pick up cone-shaped shells from reef areas regardless of whether you believe the animal is alive or dead. A recently dead cone snail may still have active venom and residual reflexive firing capability in the proboscis. The safe handling window, if any handling is justified at all, requires thick gloves and avoiding the narrow end of the shell, from which the extensible proboscis emerges during any defensive or hunting response. This advice is given and ignored constantly across reef areas worldwide, because the advice conflicts with a deeply instinctive human behavior, which is to pick up beautiful objects found on the ground and examine them. The visual beauty of the cone snail shell is not incidental to the danger it represents. It is the mechanism by which the danger reaches human beings in the first place. The shell that makes a predatory marine snail the subject of centuries of collector obsession is the same feature that causes people to ignore their uncertainty about whether the shell is occupied, and they reach down to pick it up despite whatever uncertainty they might feel. Write in the comments if you knew the cone snail was dangerous before today, because my strong suspicion is that the number of people who know this beautiful shell can kill them is significantly smaller than the number of people who have handled one on a reef without thinking about it. The gap between how dangerous this animal is and how dangerous most people believe it to be is one of the most striking mismatches in public awareness of wildlife risk that I know of, and the animal responsible for creating that gap in public awareness sits on a shallow reef seafloor somewhere in the Indo-Pacific right now, patient in the way only an ambush predator that moves very slowly can afford to be, beautiful in a way that has made its shell prized by collectors for 3 centuries, waiting for whatever comes close enough to be worth shooting at. There is one more dimension of the cone snail story that deserves attention, and it is the dimension that connects this animal to the broader history of natural history collecting. For most of European history, the shells of cone snails were traded as luxury objects, without any understanding of their biological origin or the danger associated with handling live specimens. Cabinet of Curiosities, the precursor to modern museums, routinely displayed cone shells, alongside other exotic natural objects from distant parts of the world. The shells arrived already dead in most cases because they had been prepared for trade, but the trade itself created demand that drove collection of live specimens, and some of those live specimens ended up in the hands of collectors and naturalists, who had no reason to believe a decorative shell could harm them. The 18th century Dutch naturalist and shell collector Georgius Everhardus Rumphius wrote what may be the first systematic account of cone snail danger. In his work on the natural history of the Moluccas Islands, published posthumously in 1705, Rumphius described having seen a man stung by a cone snail and dying shortly afterward, and he warned that live cone snails should be handled with extreme caution. This account predates the formal scientific classification of cone snails by several decades and represents the first documented warning in Western natural history writing about cone snail danger. It did not stop collectors from continuing to seek live specimens or from handling them carelessly. The economics of the shell trade were simply stronger than the warnings. A rare cone shell could be worth more than a year's wages for a skilled tradesman in 17th century Europe. And beautiful things that are also dangerous have always attracted human beings, rather than deterring them in the way danger theoretically should. This tension between recognizing danger and pursuing beautiful things anyway is present in every cone snail fatality involving a deliberate pickup by someone who had some awareness of risk. And it is a tension that no amount of public health warning has ever fully resolved, because the decision to pick up a beautiful object from the ground is made in a moment and with an emotional system that was not designed for rational risk assessment. It was designed for the immediate pursuit of things that look valuable or interesting. The cone snail's visual appeal speaks directly to that system, and the venom inside it is irrelevant to that conversation until after the harpoon fires. Now let's also address why the cone snail evolved this specific weapon in the first place. Because the extreme potency of fish hunting conotoxins is not accidental or arbitrary. It represents the outcome of a specific evolutionary problem that needed a very particular kind of solution. The problem was hunting mobile prey from a stationary or slow-moving platform. Most venomous animals are either fast enough to pursue and subdue prey through physical capture, or they are defending themselves rather than hunting actively. The cone snail is neither fast enough to pursue prey, nor physically capable of overpowering it. It cannot pursue a fish, and it cannot physically overpower one. The only viable hunting strategy available to an animal with this body plan was to develop a chemical weapon precise enough and fast-acting enough to immobilize prey before the prey could escape the range of the initial strike. This evolutionary constraint created extremely strong selection pressure toward 3 specific properties. Rapid onset venom. It had to work fast enough to stop a fish from swimming away in the seconds after the strike. Broad neurological disruption. It needed to shut down multiple systems simultaneously, so the fish had no escape option. Extreme potency, the dose a single small harpoon can deliver, is physically constrained. So each molecule injected needed to do more work than a larger dose from a bigger animal could. Each of these three requirements pushed conotoxin evolution toward properties that also happened to make them extremely dangerous to vertebrates generally, and extremely useful to pharmacologists and drug developers looking for precision molecular tools. The danger and the medical value are not separate phenomena that happen to coexist. They are both expressions of the same underlying evolutionary optimization toward chemical precision, potency, and rapid neurological targeting. The same pressure that made Conus Geographus capable of killing James McMichael in 5 hours is the pressure that made ziconotide 1,000 times more potent than morphine without addiction. Write in the comments if this changed how you think about picking up shells on a beach. Because the gap between how harmless a beautiful shell looks and what a living cone snail inside that shell is actually capable of doing is one of the largest gaps between appearance and reality I know of in the natural world. And the gap between how harmless a beautiful shell looks on the seafloor and what is actually inside it and what it is capable of doing in the next few seconds is one of the largest disconnects between appearance and reality in the natural world. The cone snail did not evolve its venom to be dangerous to human beings specifically. It evolved to stop a fish from escaping in the seconds after a strike. We simply share enough biology with fish that the weapon works on us too. And we share enough evolutionary attraction toward beautiful and valuable-looking objects. That generation after generation, we keep reaching for the shell on the seafloor anyway.